Welcome back, everybody, to the second episode of our BIO259 weekly recap podcast. I'm Eric, and as always, I'm here with Dr. Rosario, our resident professor and biology enthusiast.
Hey everyone, I hope you're ready because we've got some fun and important topics lined up for today. But first, let's cover some housekeeping, right, Eric?
Absolutely. So for anyone keeping track of assignments, Mastering Assignment 2 is due this Monday. And just a reminder, the deadline is 11:59 p.m., so don't leave it to the last minute.
Yeah, and if you've been having trouble with that whole pop-up blocker issue, if you're clicking the link and nothing's happening, double check your browser settings. I posted a link on our announcements page to guide you through disabling any blockers. It's worth a quick check because you don't want to miss out on getting this done.
Good tip, Dr. Rosario. Oh, and for those who might have missed the update, Lecture Exam 1 has been rescheduled. You'll want to double-check the syllabus for the exact date.
It's now happening on a Wednesday instead of the original Friday slot because of the Super Bowl. We flipped it around to give you all a little extra time because let's face it, who doesn't need more time for these exams, right?
Definitely. And, if you've got any questions, don't hesitate to reach out. Dr. Rosario, I know you're pretty good about responding quickly.
Oh yeah, either shoot me an email or make an office hours appointment with me. I'm always happy to help out.
All right, with all that sorted, let's jump into our main topic for today. Macromolecules. These are the building blocks of life. And Dr. Rosario, I know you're particularly passionate about this, so where should we start?
This is like my bread and butter. Okay, so let's begin with something I hope everyone remembers, monomers. These are the single units, the Lego pieces, if you will, of larger molecules, and when you snap a few of these monomers together, what do you get? Dimers? And then polymers, long chains made up of these individual units connecting over and over.
And what's keeping these monomers together, or taking them apart?
Great question. There are two processes involved here, dehydration reactions and hydrolysis. With dehydration, it's like, imagine connecting two pieces by removing a drop of water, click, they're stuck. But if you want to break that bond, hydrolysis is your friend. You add water back in and the bond is snipped apart. It's basically building and breaking down on a molecular scale. Super dynamic stuff.
Got it. Now, let's break this down a bit with examples. You mentioned sugars. How do monosaccharides, disaccharides, and polysaccharides fit into this?
Yes. Okay, at the very base of it, monosaccharides are your simple sugars, think glucose. They're quick, accessible energy. Disaccharides like sucrose, two sugars linked together. And polysaccharides, those are huge, like glycogen. Glycogen is how our bodies store energy efficiently. It's got this cool branching structure that lets us release glucose rapidly when the body needs fuel. It's like withdrawing multiple bills from your bank ATM all at once.
I love that analogy, Dr. Rosario. Let's turn to fats. Triglycerides versus phospholipids. What's the major difference here?
Oh, it's a key one. Triglycerides are your storage fats. They've got this glycerol head and three long hydrocarbon tails. Think butter or olive oil, saturated versus unsaturated, solid versus liquid, Phospholipids, though, are a different story. They're perfect for building barriers. They've got a glycerol head too, but only two tails. And get this, the head loves water. It's hydrophilic, but the tails, they hate it. They're hydrophobic. This is where the magic happens.
Magic? Enlighten us?
Okay. Picture this. Toss a bunch of phospholipids into water and they self-assemble into this bilayer. Heads point toward the water, tails hide inside. It's efficient. This selectively permeable barrier is what we call the cell membrane. It's super important because it controls what gets in and out of cells. Nutrients, yes. Toxins, not so fast.
Such a critical role. And Dr. Rosario, I know proteins are another area packed with complexity. Why do their structures matter so much?
Oh, proteins are fascinating. Their structure defines their function, hands down. All right, imagine this. A protein's primary structure is like a string of beads, but then it twists and folds, secondary structures like alpha helices and beta sheets. It keeps folding into these intricate shapes, tertiary structure, and sometimes they pair up into megaproteins, quaternary structure. Change the shape, and you change the job. It's like bending a paperclip. You can't use it the same way if it's misshapen.
So proteins are the ultimate multitaskers.
Exactly. They're the scaffolding for tissues, enzymes for speeding up reactions, and even hemoglobin transporting oxygen like a claw machine in your bloodstream. Insanely versatile, Eric.
All right, now that we've explored the incredible versatility of proteins, let's zoom out a bit to look at how all these pieces come together inside the cell. Dr. Rosario, when it comes to managing energy production, protein synthesis, and genetic organization, the mitochondria often take center stage. What's the story behind their nickname, the powerhouse of the cell?
The mitochondria. Let me set the stage here. You've got energy coursing through your body, fueling every single thing you do, from thinking to going for a run. That energy? It's ATP, or adenosine triphosphate, and mitochondria are the factories producing it. And these little bean-shaped structures have folds inside, we call them cristae, that maximize surface area. Think of packing a suitcase for a long trip. The more folds you've got, the more energy processing machines you can stuff inside. Efficiency at its finest.
So they're masters of energy efficiency. And am I right in thinking cells that demand more energy, like muscle cells, have a lot of mitochondria?
Exactly, Eric. It's a one-to-one correlation. High energy demand equals loads of mitochondria. Same with neurons. Your brain's energy needs are off the charts because, believe it or not, thinking requires more energy than running. It's wild, but it also shows how central mitochondria are to life itself.
All right, from energy production to protein synthesis. Let's talk about ribosomes. I love how you call them molecular 3D printers. What's their connection to genetic information?
Oh, ribosomes are the unsung heroes of the cell, Eric. They're tiny, but they're the real builders. Imagine you've got a blueprint, a piece of mRNA, which is essentially a copy of a gene. Ribosomes read that line by line and assemble amino acids like beads on a string to form proteins. But here's the cool part. These printers can either float freely in the cytosol or attach themselves to the rough endoplasmic reticulum, which we nickname the rough ER. With ribosomes plastered all over it, it's like a protein factory on steroids.
So the rough ER is key for mass-producing proteins. But what about its counterpart, the smooth ER?
Ah, the smooth ER. It's quieter, but no less important. It handles lipids instead of proteins and even has a special role in muscle cells. There it's called the sarcoplasmic reticulum, and it manages calcium. Trust me, without it, you couldn't move a finger. But yeah, no ribosomes mean no protein printing. It's a totally different department.
All right, now let's zoom in on where it all begins, the nucleus. This is like the control center, right?
Absolutely. The nucleus is like a vault full of blueprints. It holds DNA, the master instruction manual for building, well, you. And then there's the nucleolus, that dense center region. It's the factory making RNA, which is like a photocopy machine spitting out working copies of DNA instructions. But listen, the MVP here has to be histones, these little proteins that act like spools wrapping up DNA. Otherwise, your DNA would be a tangled mess, longer than a meter in every cell.
The histone's role is fascinating. It's like organized chaos. And Dr. Rosario, I know you always tie history into biology when you can. Can you quickly connect us to HeLa cells and the ethical discussions around them?
Oh, definitely. HeLa cells, named after Henrietta Lacks, revolutionized science. These were the first human cells that thrived in a lab setting, giving us breakthroughs in vaccines and treatments. But it's not all roses. The cells were taken without Henrietta's consent, raising huge questions about ethics, respect, and compensation. And yet they connect so much of modern medicine to early discoveries in cellular biology. It's a poignant reminder that behind science, there's always a human story.
Profound and humbling, really. Well, Dr. Rosario, this has been an enlightening session on the inner workings of the cell. Any final thoughts for our listeners?
Just remember, everything starts at the microscopic level. Inside these tiny spaces are the foundations of life, energy, and innovation. Keep exploring and don't be afraid to dig deeper. You'll literally find life there.
And with that, we wrap up this week's exploration of BIO259. Thanks for listening, everyone. Have a great week, and we'll catch you next time for more fascinating discussions.